Good evening, everyone. I am Dr. Jeremy Cohen. I am the director of the neuroscience program here at Xavier. I'm in the psychology department. My focus is primarily around brain imaging, and a lot of that focus then is really geared towards understanding functional brain networks and how they relate to different aspects of cognition and behavioral function. A lot of my areas of interest have sort of started around emotional regulation, emotional processing in social situations, and I would say more recently have kind of sort of merged with some of my own sort of personal endeavors with meditation. So my lab has access to quite a bit of data. We do work, somewhat closely with Dr. Jeffrey Rouse, who is in the psychiatry department over at Tulane University. So we have some different perspectives in the lab. We had a build pilot project that allowed us to acquire some functional imaging data that is both task-based and resting state fMRI that was looking at social cooperation and emotional regulatory networks in the brain. So a lot of our focus, as I mentioned in the lab, is around utilizing different computer programs, different neuroimaging processing tools. So being comfortable with computing and a comfort to dive into coding is a must. Sorry, I also have a very eager dog that is waiting by my side and is hoping for food. So he's sort of like waiting for his after dinner treat. He's being good so far. We do have, as kind of getting back on topic here, we do have a number of pipelines and procedures in the lab to get students going, that can potentially work with that data set, which still has a fair amount of unanalyzed data. We've looked, we're in the process of looking at the task-based data for that project now, but there's a host of resting state data along with different psychometric measures. We also have data available to us from the Nathan Klein Institute Rockland samples. It's an open source data set that has resting state, fMRI data as well, with a lot of additional psychometrics. Some of the ones that we're particularly interested in have focused on risk-taking behavior, impulsivity, risk perception. We even have that data set is so large that actually we've had the ability to kind of play around with some machine learning algorithms as well to try to understand characteristic brain features in that population. I'm working on a grant project now that aims to really utilize some more advanced algorithmic approaches in sort of decoding mental states associated with breath-focused meditation. And basically this is sort of built off of sort of existing literature and our understanding from a cognitive perspective of what is happening during meditation. Certainly breath-focused attention is sort of a staple and a central feature of most meditative practices. But of course, there's various stages of additional mental states in attention that shift us away from that point of focus and then ultimately take us into the experience of mind wandering, something that many of us experience throughout the day. Of course, there's also active aspects of our attention that, of course, can shift us back into breath focus. So as you can see here in the image to the right, we're really interested in using dynamic functional connectivity. And we've been working with this approach already in the lab to basically decode various mental states. And ultimately, the long-term goal of that project is actually, once we can target the state that is associated with breath-focused attention, to actually use that in real-time fMRI neurofeedback to help train individuals to become meditators more readily, more easily, and to sustain that practice. And I believe now I must open the floor to any questions if they exist.
If they have questions at a later time, would it be possible for them to contact you?
Absolutely. My email is just jcohen@zula.edu. They can email me anytime. I have office hours that they can make appointments to meet with me. My office hours tend to be on Tuesday and Thursday mornings. It's 8:30 to 11:30 on Tuesdays, and then 9:30 to 12:30 on Thursdays. But I have some additional flexibility for other meetings. So absolutely, I do encourage students to reach out and ask questions if they've got them.
Okay, thank you so very much. Appreciate it.
Thank you for your time.
Good evening to all of you, and thank you, Bill, for giving me this opportunity. My name is Navneet Goyal, and I have been trained as an organic chemist. And down the lane, I'm using this organic chemistry information to solve some of the drug discovery project. Now, what basically me and my group, and we all do, I will just give you a simple example. And I think we all are facing a very pandemic situation like COVID-19. And so many pharmaceutical companies and academic institutes are trying to find a cure for COVID-19. And we are coming up with many vaccines, but many scientists and chemists are also trying to come up with a drug. So if you get COVID, you take the drug and you're fine, right? So when you have to solve this kind of problem, you always find a cause, like what is the cause of the problem? Which particular enzyme is responsible for this disease? And then you do crystal structure docking studies, and then you design some new molecules. I'm sure many of you have seen these organic structures in organic lecture 1, organic lecture 2, or labs, right? So this is what I do. So basically, once you learn the basic concepts or basic training of how to synthesize molecule for any therapeutic area, and down the lane, you will be able to synthesize and design and synthesize drug for any kind of therapeutic area. So for example, right now I have been involved in 2 projects. One is we are designing and synthesizing the small molecules for breast cancer therapy. So as you all know, there are so many different kinds of cancer, and breast cancer is one of them. In breast cancer also, there are so many different enzymes or so many different routes. We are targeting ceramide inhibitor. And to do that, we are designing this kind of molecule. Basically, this is a ceramide analog, and we are synthesizing different kind of structures in the lab. And our synthesis is not very, very long. We are trying to avoid 10 or 15 step synthesis. So we are trying to achieve this kind of molecule synthesis in maximum 3 or 4 steps. So when you do synthesis of this molecule, you learn how to do TLC, how to do column chromatography, literature, how to present. It's a process. And once you make this kind of molecule, like ceramide analog, we do the biological assays. So we have to find out whether our molecule is active or not. So I do not do biological studies. We have Dr. Tulasi Ponapakkam, who does biological studies. and she does animal studies also. So if you join the group, you will get an opportunity to learn how to design molecules, how to synthesize molecule. And if you will be interested, you can always go and see in biology lab how they are using that molecule for in vitro, which is artificial enzyme studies, or in vivo, which is animal studies. We are doing both nowadays. So we are, so one of our molecule, which is similar to this ceramide analog, right now we are doing animal studies, and that molecule is showing some promise. So we'll see how it goes. So you can contribute in the success of this project, and we are trying to find a more strong inhibitor for ceramide. And the second project where I'm involved right now is for tobacco addiction. So we are synthesizing molecules for P450 enzyme. Now, I'm sure many of you must have heard about P450 enzyme. So basically it checks the level of, so P450 basically convert nicotine into cotinine. And when you go to a doctor and you say that I'm addicted to nicotine, or I have drug abuse problem because of nicotine, so they will check your cotinine level in the blood, and they can say whether you are in the safe zone or you are in danger zone. So P450 enzyme, what basically it does, it convert nicotine to cotinine. So if you can make a molecule which can inhibit, then you can stop the transformation of nicotine to cotinine. So right now we are synthesizing molecules which are pyridine derivative for this project. And we are doing biological assays as well in the lab. So these are the two projects I have been involved, and you will get an opportunity to contribute in one of those two projects. And I will be happy to answer any question if you have.
Well, thank you so very much. And again, as before, if you have any questions, I'm sure Dr. Goyal will be happy to take anything by email at any later time.
Thank you.
Thank you so much.
Hi, everybody. My name is Dr. Stephanie Grant. I am a school psychologist by training. And forgive me, you're going to see the desktop for just a second as I put on my presenter view. All right. So I'm here to tell you a little bit about the work that I'm doing in my lab called the JUST Lab. JUST stands for Justice in Urban Schools Together. And the primary goal of our lab overall is to decrease inequality in schools through behavioral intervention. And I'm going to tell you a little bit about the two strands of our research. Our work is kind of twofold. Okay. So first of all, I work on the school to prison pipeline. So we evaluate disproportionality interventions. So what disproportionality is the unfortunate fact that students with disabilities, and particularly students of color, are 2 to 3 times more likely to experience discipline consequences such as suspension and expulsion as students from other groups. And this is a persistent problem that we've seen in schools for over 30 years since it started being measured. And so what I'm really working on, and suffice it to say, this has a multitude of cascading negative effects. Students are more likely to be arrested, interact with the juvenile justice system, and have substance use issues, a whole manner, depression, anxiety, lack of belongingness to school. I could go on. And so my work, at least in the school to prison pipeline aspect, is really focused on a few different areas. One, we want to have quantifiable metrics of this bias in the classroom, focused on a behavioral approach. So how do we have something that the teachers are doing? Because this disproportionate discipline is not a kid problem, it's a teacher problem, it's an administrator problem. And so my work is focused on how do we change the systems and structures to decrease these negative outcomes for kiddos. And so we are actually in the process of submitting a Brady grant in order to evaluate effective interventions to decrease this disproportionate discipline experience for kids, focused on changing teacher behavior, teacher-child interaction patterns. I'm also working on what this looks like in what we call hyper-segregated schools. These are schools that are 90% or greater minority, so essentially African-American, Latinx, et cetera, and how we can improve the environments of these kiddos. And so some of the bias reduction interventions we're looking at are implicit bias training, teacher feedback, goal setting, and all of these are firmly rooted in behavioral approaches to behavior change. The other work that we're doing, maybe to some of you, is actually evaluating approaches to work with students with disabilities. So my focus is dual. I focus on students of color and how they're being marginalized in schools, and I focus on students with disabilities and how they're being marginalized in schools. One current project I'm working on right now is actually using behavioral methodology to teach appropriate social skills to students with disabilities. So we're focusing on increasing socially acceptable behaviors in order to facilitate peer interaction. And we're using behavioral methodologies akin to applied behavior analysis, if you're familiar with applied behavior analysis approaches. This is a very structured approach where we use reinforcement in order to modify behavior. And it is something that is very widely used with students with autism spectrum, developmental disabilities. So if you're interested in those sorts of behavioral challenges in kids, my lab would be a good place to learn more about how we can modify that behavior beyond ideas of maybe biological interventions like medications, et cetera, sedatives. And so one specific project that we are going to be starting data collection on in the fall, and it's I'm working very closely with a student on this project, is how do we transition repetitive behaviors common to students with autism spectrum disorder, like hand flapping, rocking, twirling, into more socially acceptable behaviors akin to fidgeting, right? So that their behavior isn't so noticeable to their peers and they're able to be more included in their environments. So ultimately, all of my work is focused on how do we make school environments more positive for the kids who are being most marginalized. And the skills that you'll gain in my lab, in addition to many others, these are just a few, are single case design and analysis, which is a methodology used within applied behavior analysis to measure behavior of individuals in which we use the person as their own control group. You'll also get practice using behavioral observation systems. In my lab, after COVID, Lord help us, we will be in schools, we will be physically in classrooms. I've already got school partners that are so excited to get our students in there and learn how to both utilize behavioral observation systems, if you're interested in behavior change research, and potentially, if you're interested, learn how to create them, right, effectively. You'll learn about behavior change process and, again, the entire field of applied behavior analysis, which is super dope. Okay, that's all I've got. If anyone has questions, I'm so excited to talk to you guys. Thanks.
Well, thank you so very much for sharing your research with us today. We greatly appreciate it, Dr.
Hi, everybody. My name is Dr. Toy Jones, and bear with me. For most folk who have had me in class or interacted with me, you know I can be just a tad bit all over the place, and it's after 5:30. So here we go. So my name is Dr. Toy Jones. I am trained as a counseling psychologist, and I'm faculty in the psychology department. So I organized my presentation to you all a bit differently, and that's reflective of the type of research that I do. So I am a part of a research collective that I have named SPARK, that we call SPARK. And SPARK stands for the Social Justice Participatory Action Research Collective. Embedded in that name is how we go about research. So we use multiple theoretical frameworks. In this collective, we work from an inter, multi, and transdisciplinary perspective. So that can mean a number of things. I'm a psychologist sitting on a team with folk coming from a variety of fields. And that very much reflects the type of research questions that we ask, as well as the folks that we walk alongside to answer some of these questions. So how we do the research. I'm going to move this over because I can't really see. So how we do the research. So in my research collective, we use qualitative and mixed methodology and analysis. And so for those of you who are psych science or pre-med psych, you've taken stats, you've taken research methods and likely advanced research. So you've learned a lot about quantitative methodology where you're using numbers to answer questions. In my research collective, we do use numbers, but there's more of a heavier emphasis on narrative, lived experience, interviewing, etc. So that's the qualitative piece. Where we do bring in the numbers is through mixed methodology. And so for us, though, in this research collective, while we do use numbers, most of our research projects, given the journals that we go after, the places where we present, and even just the questions in and of themselves, we use qualitative most often. So the participatory action and emancipatory research that we use. So participatory action is essentially saying that we work alongside community stakeholders to ask the questions that come up in our research. And I'll go through some of those variables of interest. So a community stakeholder might be someone who is the director at an agency, a social service agency, and given the nature of our research, it might also be a survivor of sexual violence. That person, along with us sitting in the collective, serves as an expert. So not only do they start the research project with us, they sit in a position where they actually inform what we do too. So this notion of expertise is shared across the board. So I might have ideas of what one's lived experience might be in terms of trauma, or in terms of trying to get grassroots mobilization efforts going. And yet the person who I'm trying to create an intervention for has the most experience, has the most knowledge. So they sit alongside us in these research projects. Ultimately, through all of the research projects that we develop, we're trying to do intervention development. So these interventions are to promote the psychological, emotional, and behavioral health of the community. And so that's always the end goal of the research that we're proposing. And so you may have noticed that I'm not necessarily presenting any specific projects. If you're interested, I do encourage you to talk to me because I can tell you about some of the projects that are ongoing right now. But given the nature of the research, we do qualitative research. So we don't necessarily start with a specific question because our endeavors are to explore what's already out there, to perhaps create something that can then turn into a quantitative research question where you have limits and numbers and it's a bit more, maybe, I don't think objective is the best word, but you get what I'm saying. So for us, the research questions is, it's something that's never been studied before. And we really want to learn about people's lived experience and then develop perhaps theory and something else that might answer what the research kind of query or question is. So the communities and variables that are of interest and focus in my research collective. So Black identified women, and that's WOMXN. Queer and femme persons, and then BIWOC communities, so Black, Indigenous, and women of color. We do some work, we do some work on with BIPOC, so Black, Indigenous, people of color, but most of our research is really addressing the everyday lived experiences of Black identified women, queer, and femme persons and BIWOC communities, so Black, Indigenous, and women of color. The variables that we're interested in, so the interrelatedness and the relationship and thinking about, in particular with these everyday lived experiences, is sexual violence, trauma, so that can be experiences, identity-based trauma, sexuality and sexual health, grassroots mobilization and acts of resistance and disruption to oppressive and supremacist systems. And so that can look like a number of things. So some of the variables regarding that last piece there that we're looking at includes things like sister care. When women of color, when Black women come together, and how that promotes psychological well-being, interpersonal well-being, et cetera. dismantling and decolonizing systems that are oppressive, and also just this notion of Black joy and pleasure and rest, so what that might look like. So these are the variables that are kind of ongoing and that are intersecting with one another in terms of the questions that we create in the collective. And so I do encourage you, if you want to know more about some of the specific research that we're doing, that you reach out. Yeah, and I think that's it. Thank you for your time, y'all.
So good afternoon, good evening, everyone. So this is Dr. Anup Kundu from the biology department. So that today I'm going to talk about the targeted delivery of doxorubicin liposomes for HER2 positive breast cancer treatment. So let me explain about some background about our research. Now, what we already know that when the cancer patients are getting treated with a cancer drug, eventually sometimes cancer cells getting develop the resistance to that particular drug, which is also known as the multi-drug resistance. So the multi-drug resistance is a serious problem to really treat many different type of cancers. And one potential reason behind developing this multi-drug resistance is the overexpression of some genes within those cancer cells. So what is our approach? Our approach is to silence those MDR genes as well to enhance the delivery of the drug to those cancer cells by using a targeted nanoparticle system. So in our research, as you see in B, in this particular illustration, so I'm showing you the pictures of the particles, nanoparticles that we make in our lab. So those are basically 100 to 200 nanometers in size, very, very small, very tiny. And it has a lot of different things. Now, as you see, it has lipids, it has polymers, but most important part is it is having a aptamer leveled on the surface. So basically that particle can really carry the drug. It can encapsulate those small drug molecules like doxorubicin. It can also encapsulate nucleic acid like siRNA, mRNA, DNA. So our purpose is to really deliver those to the specific breast cancer cells. And to do that, since we labeled our particles with aptamer, which is a specific for the HER2 receptors on the breast cancer cells, so that way the particle can carry the drug specifically to the breast cancer cells. So that is our intention basically. Now it has two objectives. As I said, we want to knock down those MDR genes. That way the drug can really get access to the resistant breast cancer cells. So the first approach, first objective is to knock down those MDR genes. And second objective is to really deliver the drug so that way it can enhance killing the cancer cells. So I'm just showing you only one slide. Now in this particular slide in number C, as you see that the breast cancer cells, which is resistant to doxorubicin. So when you are incubating cells with doxorubicin, only a very tiny percentage of doxorubicin actually getting into the cells because those cells are already resistant to doxorubicin. So then what we did, we knocked down the cells, knocked down those genes, specifically the PGP, by using siRNA delivered by our particle. And when you knock down the gene, the multi-drug resistant gene, eventually it will open up the gate so that more drug, the doxorubicin, can get into the cells. So we have been working in this area for quite some time, last few years, and we have actually published several articles on that. Most of the works in my lab actually done by my postdocs and also undergraduate students. And the second part of our project, just a brief illustration showing here. So what we recognize, like once you knock down the cells of the MDR genes on those breast cancer cells, then we apply doxorubicin as a free solution. So later we recognize like if we incubate cells with doxorubicin as a free solution, then it actually can go to everywhere. We cannot control their entry specifically to the breast cancer cells. So then what we decided, so we decided to make another set of particles that can actually hold the doxorubicin. So here is the brief illustration to really make that doxorubicin encapsulated nanoparticles. So by doing that, eventually we are targeting cells first by knocking down MDR genes, and then we are approaching the same cells by another set of particles that will carry the doxorubicin specifically. So by doing that, you actually can enhance more killing of cancer cells and less killing of non-cancer cells. So that is basically the overall objective, in my lab. Now, as I said, if you are getting a chance to work with us, obviously we'll focus this part. But since time is up, so if you have any more questions, feel free to communicate with me, or you can ask me questions that I can answer right now. Thank you.
Dr. Morgan?
Great. Well, good evening, everybody. And I'm going to share my slide here. Okay. And voila. So I'm an organic chemist, and the type of work I do is involving reaction mechanisms. And what we're trying to do is to understand how and why reactions happen, why they might happen in one case but not another, and also try to make them better so that people like Dr. Goyal, when he's making all those fancy molecules, have more options for reactions that are usable. And so I have two projects. And except for them being on reaction mechanisms, they're completely unrelated. One of them is done in the lab where you do synthesis, purification, and some rate studies. And on the other one, it's all on the computer. So they're completely different skills that you would be learning, completely different techniques. But again, the unifying theme is reaction mechanisms. The first project has to do with a reaction called the Cope rearrangement. And for those of you who have taken organic chemistry, this reaction is much like the Diels-Alder. The electrons move around in a ring and you make a new carbon-carbon bond. And as you know, organic chemists get really excited about this. This bond can form with stereoselectivity. The problem is that you have to heat it. And when you're talking about reactions with heat, oftentimes it means your molecules degrade. It costs money if you're using a commercial process. And so what I'm interested in doing is trying to figure out a way or evaluate whether we can catalyze this reaction using Lewis acids. If we have a Lewis acid hanging out here on this oxygen, what that's going to do is pull electrons out of this bond. And if you can make the bond that has to break, the sigma bond, weaker, we want to figure out how much faster the reaction is going to go. And can we make this a more practical carbon-carbon bond forming reaction for people who do complicated synthesis? And so this project, as I mentioned, would involve synthesis. We have a 4-step sequence. It would require purification and then some rate studies where you would measure how fast the product forms or the starting material goes away. Now, this type of project with synthesis, it tends to work better when there's more than one student on it. And so it's the type of thing that it works well with a group effort. And I think having those group interactions really go a long way on any project, but especially when you're working on a synthetic project, it's nice to have somebody to be there with you and kind of working with many of the same goals. The other project I have is completely unrelated, except like I said, for mechanisms. And what I want to look at is the gas phase degradation of epoxides. And these are these 3-membered ring compounds. There are a number of epoxides that are commodity chemicals. And, the problem with them is they're carcinogenic. And when they're released from factories and plants, they can cause a lot of health problems for the people who live nearby. One of the questions is, well, what's the fate of these epoxides once they get into the atmosphere? So they have this 3-membered ring that has a lot of strain, but the question is, well, how fast is that going to break apart? And what's the mechanism through which it breaks apart? Because if it degrades relatively quickly, then it's not a big deal if it's in the environment. But you know, it's helpful to know what are the options of its degradation once you form a radical here, but also, what if you put a methyl group on it? Now you have more than one radical could form. And now you have new reactions that can happen because you have different bonds that can break in the next step. And so a student working on this project would be looking at how these variations in the epoxide can change what happens once it gets into the atmosphere and once you make these radicals, which lead to the degradation. How fast do these reactions happen? How likely it is that you're going to make these different radicals, and how likely they are to degrade. And like I said, this project would be all on the computer with calculations where you would be able to discover the new reactions and look at the different options. And I'd be happy to answer any questions that you might have.
Well, thank you so much, Dr. Morgan.
Hello everyone. I hope everybody's having fun. All right, so hi, I'm Dr. Perez. I am in the Department of Psychology, and I am what I like to call myself is a behavioral neuroscientist. So I'm really interested in the brain, and I'm also really interested in how the brain handles behavior and how it interacts to influence our own behavior. My main focus really is understanding drugs of abuse and understanding the pathways in the brain that regulate the different behaviors we associate with drugs of abuse. So, quick background. So, typically we can think about drugs of abuse as being in a cycle of I take a drug, I get high. After a while, when the drug gets out of my system, I don't feel so great. So I start thinking about it, I start having cravings, so I go take the drug again. And we cycle through this, and eventually you become dependent and you can start forming behaviors that we think of as being compulsive and obsessive about continually taking this drug. We know that there are a lot of areas of the brain that play a role in the different aspects. So we know that a lot of reward-oriented pathways play a large role in the initial, I'm getting high, I am taking the drug for the first time. We know that areas that are important for stress and anxiety are important in the process in the cycle where I'm no longer on the drug, I don't feel good because I'm not on my drug. And this whole frontal region of our brain, we associate with this idea of controlling that urge and that craving and helping us decide whether we're going to go and consume more drug later down the line. So my lab is really focused on identifying pathways and how we can take these pathways that we know exist and target them so that we can target new receptor systems to make medications essentially to help reduce. Right now, we're focused predominantly on nicotine consumption, but to reduce drug consumption overall. And so currently the two main targets that we have are neurokinin receptors. So we have a lot of work that suggests that these particular type of neuropeptides are good targets to see if we can reduce nicotine consumption. And the other are these specific type of glutamate receptors, these kainate receptors, and seeing if we can antagonize those, can we also regulate nicotine consumption? So in terms of the kainate receptors, we know that they seem to play a role in alcohol abuse. So can we take, again, existing pathways and maybe use them to our advantage to also reduce nicotine consumption? So the type of questions that we are really looking at is, if we directly antagonize these receptors, can we reduce withdrawal symptoms? Because again, this is a major driver for these cravings that ultimately lead to consumption. Can we alter just how rewarding the drug is. So when I am taking the drug, can we alter how rewarding it's going to be? And ultimately, by changing these properties, are we able to actually reduce how much the drug is consumed? Now, I don't work with people. I'm not a people person, or at least not that type of scientist. I work with mice. So I do all of this work using these cute, adorable little guys. I work with C57 mice. So if you join the lab, I'm going to show you how to work with animals and how to work with mice. They're cute and adorable, and it's really hard not to like treat them as like little humanized little mice. But anyways, that's not the point. So in my lab, we use two major approaches to actually treat the animals with nicotine. Right now, the most established one is just an oral consumption. So we'll put nicotine in their waters. We are in the process of getting an e-cigarette machine, and I'm really excited about this because I really do think it's going to allow us to ask some more interesting questions, especially with the increase in e-cigarette usage that we currently see in teenagers. And so we get them addicted to nicotine. That's that first part, right? We can take this model, look and see in the presence of our antagonist, can we change how much they're either consuming orally or how much they are self-administering nicotine vapor? But we can also look and use behaviors that we know tend to focus more on this rewarding aspect. Are we making nicotine less rewarding, or maybe we're making it more rewarding during periods when the brain naturally is not very responsive to reward? The other main slew of behaviors that we do is really trying to understand all of the behaviors that are associated with withdrawal. So nicotine withdrawal in particular is really associated with increases in anxiety, increases in depressed mood, changes in ability to focus, as well as changes in our temperament. And so we have models and behavioral assays that we can take these cute little guys and see if we're actually able to alter their behavior. So if you were in my lab, we'd spend a lot of time doing a lot of behavior and playing with mice. Sorry, I have a child, so he obviously went to the bathroom. So if you have any questions, I am more than happy to address them.
Okay, this is Michelle Boissiere.
Yes.
I'm not applying for build, but I have to ask a very silly question. Who's building a vape, an e-cigarette for a mouse? And I need to see that.
So luckily, we don't have to build it. La Jolla, there's a company who builds vaporizers to give ethanol through vapor, and they have actually spent a lot of time making and creating these vaporizers that take the exact same cartridges that e-cigarette users use to make the same vapor that our teens are unfortunately smoking and using to smoke nicotine. Now, the really cool thing is once we can get this established in the lab, we can theoretically move on to other drugs of abuse. I have some work to do before we get to do that, but we do know that in general, there's a movement to use these devices to smoke other things. So it's really exciting.
All right, thank you.
You're welcome.
All right. Good evening, everybody. How y'all doing? I came with my own soundtrack. It's nobody's birthday. I have no clue why she's singing happy birthday to my mom. So first and foremost, I want to say thank you for the opportunity to present. Definitely in the midst of our current situation, these moments are important to try to maintain what we're here to do. Also want to be very intentional in saying that as students at HBCU, the need for research experience is huge, because this actually, in my profession, the reality for me is that research is oftentimes the difference maker as you continue your career ahead. So I am a licensed clinical psychologist by training. I'm an associate professor in the psych department. And it's really important that you all take this step. So I hope that you all see research here that you like, that you get involved, and that you go full on in making sure that this is a positive and a benefit to you all. And Michelle, we may need to go through Bill because they pay well from what I hear. So I'm gonna check on that if possible. So Institute for Diverse Voices is the research group, research lab that I run over in the psych department. Our kind of byline is giving voice to the marginalized and oppressed, mainly because out of the spirit of what I do personally in my practice and in the work I was trained to do, looking at groups that were historically underrepresented, misrepresented, misdiagnosed, mistreated, not treated, not included. The 4 projects that I have going on are looking at Black male experience higher education. Are there more Black men in jail or in college? That would be a question that you can find online and is asked prominently. But the point is that Black men are affected in higher education through things like identity, communication, relationships, and positive and negative experiences. So we explore what that looks like for Black men. Second project, Black female and sober. She's not a trap queen. If you look at the culture of drinking, the culture of partying, i.e. risky behaviors in college, there's a little interesting thing that seemingly happens. African-American women, women of color, don't seemingly engage the same way. And so we look at things like religiosity, ethnic, racial identity, sexual socialization, how do families, how do communities teach Black women about sex, sexual regulation, how one engages in sexual situations in general, and then just all around wellness. The third project is looking at perceptions of persons who identify as LGBTQIA. We call that what lens are you using. NIMBY, if you don't know, stands for Not in My Backyard. Basically, idea is that through the buffering hypothesis, interactional hypothesis, the more you interact with somebody who's different from you, the more likely you are to have positive experiences. So one of the questions we have is, why does it seem like people who are close to persons who are culturally different from them seemingly have more positive experiences, and then people who aren't as close, they seem to want to espouse negative views? So looking specifically at the LGBTQIA community, both on campus and in the larger community. And then a project that's really dear to my heart as a 2-time HBCU grad, and now working at HBCU, but having worked at predominantly white institutions previously, I've always wondered, why does it seem like HBCUs don't seem to provide the same provision, access, and quality in terms of mental health? So we've taken a look at HBCUs and we find that for the most part of the 109 HBCUs, only about 37, 38 of them have someone with the highest degree level over the mental health counseling identity or entity. And so we want to know, why is that? Why aren't HBCUs taking mental health care a little more seriously? Especially if we're going to be honest about it, HBCUs would be a place where a person could really develop and design a center that espouses culturally competent values, so that people of color, Black people specifically, could come into a therapy setting and feel more comfortable in terms of the seeking out and receiving of services, since we all do talk about the stigma and the demystifying of mental health care. Plus, along with that, I do a number of other evaluation things. So like, I always tend to wind up like, we're working on this, but hey, I just got this project, so let's go do that. I've done a number of evaluations in the city, for the city of New Orleans, for a couple other programs. So just depends on what's going on that we might be able to do something a little extra. So with that being said, I'll stop. That's 5 minutes. Definitely if there's any questions, holler at me, let me know. If not, my email address, as well as the website, which actually lists a lot of the things I do that I incorporate into research, is there as well.
Thank you so very much.
Thank you.
A big thank you to all of our faculty for taking the time out to present, and also to the students for coming to hear about all the great research we have at Xavier. We really appreciate everyone's time.